We'll actually be beginning Lecture 8 with a discussion of a few final topics for metabolism and catabolism specifically. We've been using cellular respiration as an example for these catabolic reactions, but it's not as linear as we've actually taught it. It's a tightly regulated and controlled process, and it's one of only one possible catabolic reactions that a cell could use. Take a look here at this metabolic map for a human cell. You'll note that there are many different reactions which the cell can use. In addition to the fact that the cell's core citric acid cycle here is being fed by glycolysis, there are other components which can feed into this central reaction, as well as ways that we can actually utilize these processes coming off. If we take a look at this process, there's a few things I want you to take note of more specifically. The first is that glucose is not the only fuel that the cell can use. Other sugars, other carbohydrates, which the cell is capable of bringing in, such as starch and glycogen, can go through the process of glycolysis. If we utilize other components like proteins, or more specifically the amino acids, once the protein is broken down, then we can feed into different components. So we might actually feed in a 3-carbon molecule as a substitute for pyruvate, or a 2-carbon molecule, which is being broken down and modified to become acetyl-CoA. And then side chains can also feed into other places. Fats, which can be broken down by beta oxidation into glycerol and fatty acid, can feed in as G3P, the 3-carbon molecule, or the fatty acid tails can be broken down into 2-carbon molecules, which yield acetyl-CoAs. This process of actually creating these individual carbons from beta oxidation yields more ATP per gram than sugars. This is one of the reasons why your body chooses to store extra energy as fats that it can break down later on. Now, some of these intermediates can also be used for other processes as building blocks for other components. The example which you see here is the NH3 amino group, which is broken off of amino acids that can actually go to be utilized, for example, as a component of nucleotides. But there are many, many other examples of this process. I just expect you to know that this is a possibility, not know any specific pathways. However, you should know that each one of these pathways is very tightly regulated and controlled. Metabolism is all about cellular energy, and so it shouldn't surprise you that the cell only wants to expend energy when it's necessary, and it wants to conserve it wherever it can. A great way for the cell to do this is through the use of feedback inhibition, which hopefully you recall is the process by which cells utilize end products to inhibit earlier stages in a pathway. An example is the use of ATP to inhibit the enzyme phosphofructokinase. One of the enzymes which you need to know is a component of glycolysis. When there's lots of ATP in the cell doesn't need cellular respiration to produce more. And so lots of ATP being produced can actually inhibit the activity of phosphofructokinase. As a counterpoint to this, lots of ADP, which is going to be an expended component, right? When we break down, we hydrolyze ATP, it yields ADP. So if we've used most of our ATP and we have lots of ADP in the cell, will actually activate phosphofructokinase. This ability to be either activated or inhibited is because phosphofructokinase, it can be allosterically regulated, which allows these two components to be in opposite of each other. So here's a question for you on Socrative. Which level of protein structure is disrupted by allosteric inhibition? Pause here if you need more time. Good, this is quaternary level of structure. Hopefully you remember and recall that if quaternary level of structure is 4 individual, or in this case 4, you can see a picture of phosphofructokinase with our 4 individual polypeptides, each in a different color, that we can use a molecule of ADP to activate and stabilize this quaternary level of structure, or molecules of ATP to inhibit or destabilize this structure. Now we can also look more closely at types of metabolism that are used when there is no oxygen present. So far we've been looking at aerobic cellular respiration, where the process of the electron transport chain is really being driven by that highly electronegative oxygen accepting our low energy electrons that have been used to pump protons. But without oxygen present, the electron transport chain ceases to operate. If this is your only option, you're an obligate aerobe. On contrast, if you are capable of utilizing other processes, such as obligate anaerobes, which do not use oxygen and they cannot survive in the presence of oxygen, or facultative anaerobes, which can use either fermentation or aerobic respiration. Now, note that I didn't say aerobic or anaerobic respiration. That is not going to be a normal or common pathway. It's actually extremely rare. But being a facultative anaerobe is very, very common. These are organisms that use aerobic cellular respiration when oxygen is present. This allows them, in the presence of oxygen, to generate maximum ATP, 32 ATP per glucose. But when that oxygen is gone, they can very easily take the glycolysis that they're already doing, and in the absence of oxygen, no oxygen needed, regenerate the NAD that's needed for glycolysis and just make 2 ATP at the same time. So facultative anaerobes are capable of utilizing aerobic cellular respiration, but when oxygen is absent, switching over to fermentation. Fermentation has one single goal, and that is simply to remake NAD+. You'll recall from glycolysis that during our energy payoff stage, that we actually need to utilize a molecule of NAD+ to accept a pair of electrons so that we can use our triose phosphate dehydrogenase to create the next step. This means that this is a reactant. Whether we think about the fact that NAD+ needs to be utilized to do something later or not, NAD+ is necessary. The problem is that in the absence of oxygen, there's no use for NADH. Remember, we have no electron transport chain for NADH to donate electrons to. So the entire purpose of fermentation is to take a molecule of NADH and oxidize it back to NAD+. Fermentation reactions will serve as another option for NADH to be able to donate its electrons. And depending on the type of fermentation, it'll depend on the type of reaction. Alcohol fermentation, which is used in yeast making or used by yeast in bread making, brewing, or wine making, is going to use acetaldehyde as the molecule that accepts these electrons and yields a product of ethanol. That's why alcohol is being produced. Lactic acid fermentation, such as the one that's used in your muscles when you run out of oxygen because you're doing anaerobic exercise, would be something that uses acetaldehyde's byproduct and yields lactic acid at the end. So tell me in Socrative, what is the purpose of fermentation reactions? Pause here so you can have more time. Good. The purpose of fermentation reactions is to regenerate NAD+, so that glycolysis can continue. Fermentation produces 2 molecules of ATP, because that's the net amount of ATP that's being produced by glycolysis, and that's the entire amount of ATP that was produced. There is no use for alcohol or lactic acid. It's a toxic byproduct. It's waste product for the cell. It's not trying to make alcohol or lactic acid to be used in any other way. It's not buying time. It's just allowing the cell, even if it's a tiny amount of ATP, to be able to continue to do glycolysis. So tell me in Socrative, Facultative anaerobes can utilize which forms of cellular catabolism? Select all that apply. Well, you know that facultative anaerobes are going to be doing aerobic cellular respiration because that's going to be the most efficient form. But are they going to do anaerobic cellular respiration or fermentation when oxygen is absent? It's going to be fermentation. There are an extremely few number of organisms on this planet that are capable of switching between aerobic and anaerobic cellular respiration. Now what I'd like you to do is to pause the video here and to go forward with the next 15 questions so that you can review and see how much it is that you know about cellular respiration before we continue. There's a number of short answer questions and some fill in the blanks. Really try to test yourself and see how you're doing. And after you've answered question 18, then come back here and I'll go through the answers. So how many NADH produced per glucose in aerobic cellular respiration? Well, you get 2 in glycolysis, 2 from the oxidation of pyruvate, and 6 in the citric acid cycle. So that means that there are 10 NADH total, 2.5 ATP produced per NADH. And since this process is not directly coupled with the process of creating ATP, this wouldn't be a whole number. Remember that the process actually involves the pumping of protons. NADH is going to donate its electrons to an electron transport molecule in the electron transport chain. And it's those molecules that pump protons, and then it's the proton motive force that's actually responsible for turning the rotor in ATP synthase. FADH2 is donated later in the chain and pumps fewer protons, so it results in the production of less ATP. 1.5 per FADH2. And when you described to me the process of the citric acid cycle, did you start and end at the same step? Meaning, if you mentioned oxaloacetate as the first molecule, did you come all the way back to mention oxaloacetate again? Did you describe how it combines with acetyl-CoA to form the 6-carbon citrate? Whatever process in this you started with, you need to end with. And you need to make sure that you mentioned all 3 of those molecules. Now, following the citric acid cycle, the majority of energy from glucose is found in these electron carrier molecules because they're the ones that accepted the electrons and became reduced. Because what we did was oxidize glucose in a series of redox reactions, reducing the electron carriers as we went. The purpose of the electron transport chain is to pump protons into the intermembrane space. There's no point in taking all of these electrons just to donate them to oxygen at the end, because the production of water doesn't do anything for the cell. We call this pumping of protons, the resultant force that it creates, the proton motive force. It's a very special electrochemical gradient which is in the cell, and we give it a special name because it can do special jobs. When those protons flow back down their concentration gradient through the rotor of ATP synthase, that's when we finally catalyze the production of ATP. And because oxygen is responsible for keeping the electron transport chain flowing, ultimately it's responsible for keeping the entire chemiosmotic and oxidative phosphorylation process going. Now, how many carbons would this molecule have? If you were to take a look at oxaloacetate, you would see that it has 4 carbon molecules contained, 1, 2, 3, and 4. Acetyl-CoA has 2, which means that this molecule has to have 6 carbons. And we know that the 6 carbons belong to citrate, or citric acid. When you take a look at this step of the citric acid cycle, we'll notice that the first one here, that this molecule, which is a rearrangement of citrate, has 6 carbons. These 6 carbons become oxidized with the loss of carbon dioxide to form a 1, 2, 3, 4, 5 carbon molecule. And since we've oxidized this molecule to lose a carbon dioxide, we had to reduce a molecule of NAD plus to NADH. In step 4, we go from a 5 carbon molecule to a 4 carbon molecule. And this results in the loss of CO2. Well, since we've oxidized this molecule of CO2, we also must reduce a molecule. And so we're going to go from NAD plus to NADH. In this step, you'll notice that we went from a molecule that's 4 carbons to 4 carbons. So we didn't lose a carbon dioxide. And that also means that we didn't reduce a molecule of NAD plus to NADH. But you will notice that there's an inorganic phosphate here which is being attached. GDP becomes GTP, or you could also say that ADP becomes ATP. What type of phosphorylation did this? Well, there's no oxygen present or necessary here, and we aren't doing photosynthesis, so this would be substrate level phosphorylation, the creation of an ATP molecule catalyzed by an enzyme. Now I want you to pause the video here and try answering these 2 short answer questions. Describe the process of oxidative phosphorylation, and then why do humans breathe out CO2? You should be following these guidelines while you answer the question. On the exam, I won't give you these guidelines, but I would expect any short answer question to be answered using these steps and including this information. And then I'll see you in the next video where we'll start to discuss the process of photosynthesis.